Okay, our next speaker is Dr. Hervé Falet, who is joining us from the Versity Blood Research Institute in Milwaukee, Wisconsin. Hervé completed his PhD at Paris Descartes University, followed by a postdoctoral fellowship at Harvard Medical School, where he stayed on as a faculty member for many years. So I first met Hervé when I went to John Hartwig's lab for my own postdoc, and Hervé, by definition, is the person who knew me back when I was young and stupid. But that's I suppose a separate issue. So Hervé has an encyclopedic knowledge of platelet biology, and he certainly taught me and many others a lot about how platelets work. And I think it's a testament to that many of us are still working on platelets today. So we're very fortunate to have him join us today. He'll be speaking about his work related to his interests on interactions between the platelet cytoskeleton and the plasma membranes. Hervé, welcome.
Hello, thank you for the introduction and for the invitation to come here. It's a pleasure to be at this symposium. I wanted to be here for quite some time, and I'm glad to be invited today. So one of the focus of our research is platelet production by megakaryocytes. So as you heard from the previous talk, it takes from HSCs to mature and differentiate to form a megakaryocyte. So it goes through increased ploidy and formation of a membrane system, which is very particular to megakaryocyte, called the demarcation membrane system. And so studies have been done for quite some time on this structure, and it's known from the work of Anita Eckley on the left, but it starts with a single invagination in the megakaryocyte, and then with maturation of megakaryocyte, it occupies the whole cytosol, but always being connected to a plasma membrane. It's an invaginated system. So one of the big questions in our lab is what are the proteins and mechanisms involved in the formation of a demarcation membrane system in megakaryocytes. So the outline of the talk, I will do a brief introduction on filaminae, which is a cytoskeletal proteins I worked on for quite some time, a long time ago. Cytoskeletal scaffold protein, which is required for platelet production and function. I will then move to the role of Paxin-2. So filaminae binding protein and membrane bending protein in demarcation membrane system formation in megakaryocytes. And finally, because it's a hemostasis symposium, go more into the role of Paxin-2 in platelet hemostatic function. So filaminae, yes, that's a protein, or large protein expressed in platelets. It's form of an actin binding domain at the N-terminus. If I can use a pointer. Then there are 24 immunoglobulin repeats of 400 amino acids each. So repeats 1 to 15 form this long arm, and then repeats 16 to 23, a more compact structure. That's where the action happens. That's where filaminae binds to platelet glycoproteins such as GPMB alpha and beta integrins, and then repeat 24 is the dimerization domain. So filaminae cross-link actin filaments. It tethers membrane glycoproteins and also serve as a scaffold for signaling intermediates. So in platelets, filaminae links the VWF receptor subunit GPMB alpha and beta integrins to the cytoskeleton. GPMB alpha binding to repeat 17 and integrins to repeat 21, but we'll discuss that in the talk. So the gene encoding filaminae is on the X chromosome in both humans and mice, and there are filaminae mutations in humans. They are usually embryonically tall in males, and you have brain malformation in heterozygous females and also thrombocytopenia, but which is very kind of difficult to see because most of these platelets are actually platelets are mosaic, and 95% of the platelets are usually positive for filaminae in female patients. So we have generated mouse models for filaminae deletion in platelets, and these mice display an increased thrombopoiesis in vivo. They have a severe microthrombocytopenia, so very large platelets, and not too many. And that's due to accelerated clearance by macrophages. They also have a bleeding diathesis. So platelets themselves, now they have a diminished cytoskeletal integrity, and you can see that from this EM on the right. You remove a membrane of the platelets, you look at the cytoskeleton by EM, you can see nice actin filaments on the left. In the right, platelets are larger, and they last these cross-linked actin filaments that you see in the wild type. We also have decreased expression and altered distribution of GPMB alpha, which result in decreased adhesion to collagen at high shear. And also what we observed in that studies, there was also impaired GP6 and CLEC2 signaling, which resulted in decreased hemostatic function due to the loss of the filament-Syk interaction. So back to the demarcation moment system. Now we look, compare wild type and filament knockout, and you can see in the wild type, you have nice platelet territories in the cytoplasm of a megakaryocyte. Now platelets are nice distributed with a gap between them. We call that platelet territories. It's missing in the filament knockout. So filament is required for demarcation moment system formation in megakaryocyte. But filament itself does not bind to membranes. So we look for partners who could bind to both filament and to membranes. And we focus on one family of protein called the bar proteins. And particularly one of them, the bar proteins contain 70 proteins. So we focus on one called paxin-2. Paxin-2 is implicated in endocytosis, particularly caveolae biogenesis, endosomal trafficking, cell adhesion, spreading, and migration. And out of the 70 bar proteins, that's the only one, paxin-2 is the only one known to bind filament. So that was an interest right there. So filament binds to the F-bar domain of paxin-2, and paxin-2 also have an SH filament at the C terminus. It can bind to endocytic and cytoskeletal proteins such as delamin-2 and NOAs. And important for human platelet biology, there are single nucleotide variants in humans associated with decreased platelet counts and increased platelet size. So what we found in collaboration with a group of Walter Carr in Toronto, Go Canada, we see paxin-2 associating with a single invagination in megakaryocyte, what Anita Eckley previously described as the initiating demarcation moment system. So we look at the interaction between filament and paxin-2 in vitro using purified proteins, and we were able to find that paxin-2 through its F-bar domain binds to the tip of the F-bar domain, so the two tips of the F-bar domain bind to repeat 20 on filament. And somehow we came up with that model where filament, because both filament and paxin-2 are flexible dimers, that filament binding to paxin-2 could regulate membrane tubulation activity. And we were able to see that in vitro using liposomes. We saw that also in platelets. And I will show you also in megakaryocytes. So if you have GFP paxin-2 expressed into wild-type megakaryocytes, you see a nice imagination in the cytosol. If you mutate the F-bar domain so it doesn't bind filament, or if you express wild-type paxin-2 into the filament knockout, you lose that structure. So since then, we have obtained the paxin-2 knockout, originally the full knockout, and more recently a Flox mouse, which we bred with PF4 to obtain megakaryocyte-specific knockout. And both mice have a very similar phenotype. They develop a mild thrombocytopenia with increase in platelet size. Looking at the DMS structure, very similar to what we observe in filament knockout. You don't have a nicely formed demarcation moment system in the paxin-2 knockout. Very similar to what we observed before with filament. And the object of our studies right now in the lab is looking also at another structure that megakaryocytes do when they form platelets, this polosome peripheral zone. So that's the structure that is required for megakaryocytes to pass the endothelium to be able to release platelets into the vasculature. And in the absence of Paxin-2, we observe less of this structure and also a less dense structure in the one that do contain the structure. So in summary, that first part, the F-bar protein Paxin-2 is an internal marker of the initiating demarcation moment system from, in megakaryocytes. Filamine binding to Paxin-2 F-bar domain regulates membrane tubulation, both in vitro in platelets and megakaryocytes. Paxin-2 knockout display, mice display a mild thrombocytopenia with increased platelet size, which is consistent with variants observed in humans, and Paxin-2 knockout megakaryocyte display impaired demarcation membrane system, polosome peripheral zone formation, as well. I didn't show you today, but we have also reduced ploidy and decreased proplatelet formation. So moving to the hemostatic function of these mice, we first do a tail bleeding time assay. You cut 2 millimeters of the tail of the mouse, and you observe how long it takes to stop bleeding. And when you can observe from the blue line compared to red, there is a delay in bleeding time in the Paxin-2 knockout mice. We move to a thrombus formation assay. The ferric chloride induced carotid injury, and again, very similar. You see a delay in thrombus formation in the Paxin-2 knockout. Because it was a full knockout, we to prove that, to show that it was specific or intrinsic to platelets, we took wild-type platelets, injected into knockout mice, and we normalized the thrombus formation. So again, indicating that the phenotype was intrinsic to platelets. We haven't done, we just got the flox mice. We haven't done these experiments yet in the flox model. What was very striking was really in another thrombus formation model, the laser injury mesenteric model. First on the left is the wild type. You can see accumulation of platelets in red and fibrin in green. You have a nice accumulation of platelets and goes down slightly over time. If you do the same experiments in the Paxin-2 knockout, the thrombus form but falls apart. Form falls apart. That's like very regularly you have severe embolization of a platelet thrombus in the Paxin-2 knockout. Here's the quantification of the data. Now again, on the wild type, you see nice accumulation of platelets. It goes down over time slowly. In the Paxin-2 knockout, every 15 seconds, like a clock, it goes up and down. By contrast, fibrin accumulation was not affected. So, because as you said, I know we've done platelets, I say, at least the one that told him. Or maybe he told me, I don't know, I'm not sure what. Anyway, so we do platelet activation, stimulating platelets with different pathways. One which is GP6 with a collagen-related peptide, which goes through phosphotyrosine, Src kinase, SYK. Another one which is thrombin that goes through protease, activated receptors, which are G-protein coupled. In both cases, we looked at fibrinogen binding as a marker for alpha-2b beta-3 activation. We also look at p-selectin expression, and you can see from this data there was no difference in activation between wild type and knockout. Another experiment we've done was aggregation, here in response to collagen, and again there was no difference between wild type and knockout. So I mean, that was disappointing. So how can you come from, you see this severe embolization phenotype, which is platelet intrinsic, and there is no real phenotype in platelet function. So we looked at another response. One was the spreading. We used two different ligands on the surface. One was fibrinogen, which is specific for alpha-2b beta-3, and one was a peptide called GFO-GER, which is specific for alpha-2 beta-1. And as you can appreciate from the data on the left, and also the quantification in the middle, is that you, in spreading on fibrinogen was not affected by Paxin-2 deletion. By contrast, spreading to the peptide for beta-1 integrin was increased. So we check for integrin beta-1 activation. We use flow cytometry using an antibody called 9EG7, which is specific for beta-1 integrins. And we could see that in PAXIN2 knockout, yes, there is increased activation of beta-1 integrin. By contrast, there is no change in the expression of beta-1 itself. So what do we know about beta-1 integrin in platelets? There are at least three, depending on which alpha subunit is linked to, alpha-2, alpha-5, and alpha-6. And the work on beta-1 is quite confusing, because if you look at the data that goes back to 2000 from the work of Bernard Nisland, if you do a knockout of beta-1 using the MX1 or more recently using by Pierre Mangin and Strasbourg using the PF4 there is not really a role for integrin beta-1 in hemostatic function. By contrast, a more recent study by the group of Karen Hofmeister, I showed that if you have a difference in glycosylation on beta-1, and that leading to activation of hyperactivation of beta-1, what you see is thrombocytopenia and bleeding. So the question we had was, could it be that the phenotype of the PAXIN2 knockout, could it be driven by hyperactive platelet integrin beta-1? So to test that hypothesis, that question, we bred the PAXIN2 knockout in the integrin beta-1 knockout. So we bred PAXIN2 full knockout with a platelet-specific beta-1 integrin. And as you can see from looking at the platelet counts first, there is the integrin beta-1 deletion normalize the platelet counts of the PAXIN2 knockout. So there is a role already in thrombopoiesis of beta-1 integrin. But then if you move to this hemostatic assay, looking at bleeding time, or the ferric chloride-induced carotid injury model of thrombosis, you normalize the phenotype. So deletion of beta-1 normalize the phenotype, the delay in bleeding time and thrombus formation we observe in the PAXIN2 knockout. But also, which was, I think for me, even the most striking was looking at the laser injury mesenteric model. Again, you normalize the phenotype. You don't see the severe embolization that you observed before in the knockout. So how do we link PAXIN2 to integrin beta-1? And I'm back to filamin A. Now I show you in the first part of the talk that PAXIN2 binds to repeat 20 on filamin A, which is close to the repeat 21, the integrin binding site. If you look at how ligands bind to filamin A, like for example GBNB alpha to repeat 17, or integrin beta-7, or beta-1, or beta-2, which was published to repeat 21, the structure looks the same. So the binding is very similar between the two. However, when you look at the structure of repeat 16 to 21 on filamin A, you don't have a linear structure. You have a propeller structure with each blade of a propeller being composed of 2 repeats of filamin A. So you have 16, 17 together, 18, 19 together, and 20, 21 together. But quite interesting, if you, for example, 16, 17, the 2 repeats are back to back. The binding site for GPMB alpha is open. And that's we know, I guess Rose knows that GPMB alpha binding to filamin A is constitutive in platelets and megakaryocytes. By contrast, if you look at integrin, there is a beta strand in repeat 20 that covers the integrin binding site on beta-1. So for integrin to be able to bind to repeat 21, you need a conformational change of that structure. So we came up with a model because PAXIN2 binding to filamin A to repeat 20. We asked, can PAXIN2 binding to filamin A repeat 20 modulate integrin beta-1 binding to repeat 21? So to test that hypothesis, we incubated beads coated with the bound to the integrin beta-7 cytoplasmic tail. We incubated these beads with filamin A repeat 16 to 23 in the absence or presence of a PAXIN2 peptide that binds to repeat 20. And as you can see from this blot on the right, if you don't have a peptide, the integrin beta 7 cannot pull down filament repeat 16 to 23. You incubate with a peptide, then there is binding, indicating that Paxin 2 binding to repeat 20 can change the conformation of the repeats, allowing integrins to bind to filament A. By contrast, if you take repeat 21 alone, so there is no repeat 20 covering the binding site, the peptide has no effect. There is binding no matter what. So in conclusion of that second part, now Paxin 2 knockout mice display mild bleeding diathesis, which is due to severe thrombus embolization. Paxin 2 knockout platelets display increased activation of integrin beta 1, but activate integrin alpha 2b beta 3 and secrete normally. If you delete platelet integrin beta 1 deletion, normalize the hemostasis and also platelet counts in the Paxin 2 knockout mice, and also in vitro, Paxin 2 can mediate beta integrin binding to filament A. So we came out to that model where there is a bidirectional interaction between filament A and Paxin 2. In one case, filament A binding to Paxin 2 regulates membrane tubulation by the F-bar domain of Paxin 2, but also Paxin 2 binding to filament A regulates how integrins get activated. So in the presence of Paxin 2 binds to filament A, allowing binding to integrins. They cannot be activated by kindlin and tallin. If you don't have Paxin 2, then the tail of integrin is open and can be easily activated by tallin and kindlin, leading to hyperactivation and the bleeding phenotype we observe in the Paxin 2 knockout mice. And with that, I would like to thank my colleagues, collaborators, funding agencies, and you for attending. Thank you.
That was a nice talk, Hervé. So the model that you put up of filament A, it's a dimer and it's kind of bent in a V, and that V is clamped together by the Paxin 2. You would think that that would change the spacing between binding sites for the membrane proteins on the two dimeric units. And so if you don't have that thing there, maybe it opens up, and now say between one GP1B alpha and another GP1B alpha, the distance is greater or less or something. And that could be true of the beta integrins. So I'd like you to speculate on that, and whether you have looked at VWF-dependent platelet adhesion in your knockout.
Yes, we've done, and we see a defect as well. So they have a decrease in adhesion to collagen under high shear, so which is GPMB alpha dependent. So we know that from the
I don't know if you could do like a resonance energy transfer experiment to see if the distance between different subunits changes.
Yes, I mean, that's a nice model. I like to show it during presentation like that, but I'm not sure that's correct. I don't know. No, I don't know if that's really intra or that's inter. That could be a chain. I mean, I also see that as a zipper somehow. That you have filament A Paxin, filament A Paxin, filament A Paxin. That could be another way to do it as well.
Yeah, well,
that's one thing I would like to do is to get the structure of that. I would like to see that as
But the structures are static, and you just showed that it's not static.
Yeah, I mean, we tried badly to get the structure of repeat 2021 with a Paxin 2 peptide. You can crystallize it. It's non-stop moving.
And I'm going to also warn you that once you draw these models, they become kind of fact.
I'm sorry, then.
Very beautiful work. So what do you mean the beta 1 integrins? Is alpha subunit or beta subunit interact with that portion?
Yes, I mean, that's a big question we have as well. Which integrin beta 1? I mean, I can speculate if you want. I made 2 slides. I guess that's for you. No, So yeah, there are three alpha subunits we know binds to beta 1. Alpha 2, which binds to collagen, alpha 5 to fibronectin, alpha 6 to laminin. Of all the three, fibronectin is the only one soluble that exists as the soluble form in plasma. So that's one we focus on. To be frank with you, I was expecting the defect in the amount of soluble fibronectin in platelets, but there is no change. So it seems endocytosis of fibronectin is not affected by the lack of paxin-2. By contrast, cellular fibronectin was decreased by half. So some paxin-2 non-gut megakaryocytes make less fibronectin than wild type. So that indicates somehow that's alpha 5. That's something we plan to investigate. Now, we just got the alpha 5 flux mice. We tried to breed them with paxin-2 and repeat these experiments. What we've done with beta 1, do it with alpha 5 to see if that's alpha 5. That's where we start. We think it's alpha 5. If that's not, we'll move to alpha 2 and alpha 6.
I do appreciate that you prepare this slides for me, but you probably also keep in mind that plasma fibronectin could be, majority of the platelet fibronectin is internalized. 80 to 90% of them is internalized by integrin.
Yeah, which
So
could be
that-
alpha 5 beta 1 or alpha 2 beta 3. But I think what you've shown in your papers now with Denisa Wagner.
Right. So therefore, whether this is because of internalized by beta 3 or beta 1, so that's maybe still more work could be done. Yeah. Thank you.
Yeah, sure.
Sorry, can I broaden the base a bit? You've got an X-linked protein, and you saw what happened when you had a mutation in the mouse that was lethal. Was that an induced mutation, or do these mutations exist in the human population spontaneously? Because then it raises the question of lionization, where you could validate this by studying the human population. Does that make sense?
You're talking about paxin-2 mutations or beta 1 mutations?
You had a mutation in your...
That's a
male,
knockout, yes.
And
Yeah.
then it was lethal. Okay, fine. So then, do these mutations exist in a human population? Because if they do, then lionization could give you a level of diversity to test your hypothesis.
So there are, I don't think there are full deletion of paxin-2 in humans, but there are variants. And that was done in the UK. There was some nice genetic studies or genomic studies. And paxin-2 shows up quite often with p-values of 10 to minus 200. I mean, that's there is very strong association between paxin-2 variants and platelet count and size. I don't know if people know. That's something I would like to do, actually. That's something I would like to move to the human, not just use the mouse model, but also see how it affects human population.
Thanks.
Okay, thank you. Oh.
I'm curious, further to Jose's hinging question, I'm wondering if you've checked whether the filament has been proteolyzed, like by its known proteolytic cleavages that calpain may induce to facilitate paxin interactions.
I looked. I didn't see any proteolysis of filamina in the paxin-2 knockout. No.
Thank you.
Yeah.
Thank you very much.